So let's start the session. So now, as yesterday we had good presentations on the genomic resources, genomics platform, and today is the day for the breeding, integrated breeding, genomics assisted breeding. So without taking any time, I would like to invite Dr. Ramesh Agrawal from CCMB Hyderabad and Dr. Estefania Grando, Research Program Director from for Dryland Cereals from ICRISAT to co-chair this session. So please join me in welcoming both of them. And now session is over to you both. Please start. Thank you very much.
A very good morning. We welcome you all to this first session of the day, and the session is on genomics assisted breeding. You will agree that yesterday we heard about the great advancements which have happened in the last few years in the tools and NGS technologies, which has also led to generation and availability of huge amount of genomic data for a large number of crop species. And today we hope to hear from various speakers in this session and other sessions on the day about the path forward, how to basically, for considerations like how to deal with this huge amount of generally unstructured genomic data, and how to use this for plant improvement or breeding. Today in this session we have 3 speakers, and I appeal to them, please try to complete their presentation in 25 minutes, the time allotted to them, so that we have some time for the questions. And before initiating the proceeding, I would like to thank the organizers of the meeting to invite us to this meeting and also to ask us to co-chair the session. And now I invite Dr. Visser to make his first presentation on possibilities and limitations of plant genome sequences for plant sciences. Go.
Okay, thank you very much. Good morning, everyone. I would like to thank Rajiv for the invitation to come here and to give a presentation. Have to stay in time. So what I would like to do this morning is to go through a couple of things which I think are, from a breeding perspective, important in relation to all the sequencing things which are coming our direction. So I will tell something about breeding and sequencing, something about genotyping by sequencing, which we have done recently in tomato, then something about genomics and gene banks, and then a few things about, let's say, what I think is important to create. The virtual lab of plant breeding is one of these, BreedDB, a database, and an issue which I term to call precision breeding. So as you can see, and as we also have heard yesterday, and you are able to read in many journals, almost every week there will be a new species sequenced. So we have a lot of sequence information, and then the big question, of course, as a breeder is, and now what? What can we do with these sequences? And you can look at it from different sides. You can say, okay, this is a genome sequence. Well, of course not, because you only sequence one individual, maybe even one haplotype in many cases. Sequencing one genotype is sequencing not a species, so you still have a lack of information about your desired genotype. On the other hand, you can also look at it from a positive point of view and say, okay, I have now a sequence, and at least I can design primers, and I can try to focus on my favorite region and maybe sequence only that region and get more information about my desired genotype. But in order to get a better idea about what is going on in genotypes, or rather haplotypes, we need more sequencing. And then of course you can say, okay, do we need to deep sequence more, or do we need to sequence more different individuals? Well, if you look at genomics and at orphan crops, then I think it's a blessing that we have genomics. With a few cost and research efforts, quite a lot of information in ornamentals like tulip, but also in a lot of orphan crops like yam, papaya, etc., etc. You can relatively easy find and after confirmation use SNPs, so that is quite good. And of course you can also use the comparative genomics approach for known pathways in, for instance, fruit development or in color, so that you can use that in your particular crop of interest. And what I think is most interesting, and that is one of our focus points in Wageningen, is that sequencing and polyploids now make it possible that breeding in polyploids can be sped up, can be increased, can be made faster. But if you have the sequence, then what are you going to do with it from a breeding point of perspective? How useful is the sequence really in breeding? Because next to, let's say, the bare sequence, we also have a lot of other omics technologies like transcriptomics, metabolomics, and we need to, or those kind of data is assessed, and once you have the data, you have to store that, and you have to design tools in order to be able to use those kind of data. We already heard it yesterday, is that there is a lot of need for better phenotyping, because that is the bottleneck at present. You need to phenotype your genotypes extremely well for your traits of interest. Of course descriptive traits, but also the metabolomics, proteomics, and what is needed at the moment is automation of those kind of analysis, because it's much too much time consuming. So if you look at breeding, then let's say for the last 100 years it has been an art. Genetic improvement has been done mainly by phenotypic selection. Progress is very slow. You need what is called green fingers and breeder's eyes, and if you have, or if you happen to stumble upon an excellent variety, it mostly is by chance. And this chance, of course, has to be improved because now we want to breed more for specific markets with a much higher and even predictable chance of getting excellent varieties. And I think genomics will enable this. And that means also that breeding becomes more technology based, and therefore I'd like to call that precision breeding, and in my last few slides I will spend a few minutes on that. So the key message is you have to phenotype and genotype your crossing parents, because if you know which parents you want to use in your crossing experiments to make new genotypes, to make new varieties, then you need to know them inside out, and then you can much better predict what is going to happen. So we've done some genotyping by sequencing in tomato, and we already heard it that sequencing costs are coming down and down, and you can ask any PhD student, the first thing he or she would like to do is to sequence something. But the question is, well, is it wise to sequence? What should you sequence? Why do you want to sequence it? So of course you can spend a lot of money on sequencing, but the issue, at least for me, is what do you want to do with the sequence? So what we did in this experiment, and this is part of the 150 tomato genome project, is that we took a cross between moneymaker, a variety, and a wild species, Pimpinellifolium, Solanum pimpinellifolium, and from that we made recombinant inbred lines already a long time ago, and from about 100 F2 plants we made the single seed descent, and we got homozygous plants. And of those, about 60, 65 were sequenced. And well, if you take a look at the sequence of all these recombinant inbred lines, then of course there are a number of questions you can ask, and one of the questions we were interested in is it possible to determine the boundaries of the introgressions of the wild species into the variety? And of course there are a number of challenges because we did the sequencing at a relatively low coverage, so we have quite a lot of missing calls. We need to do some filtering as shown here, but in the end of the day it was possible to get these kind of pictures. What you see here are a number of these recombinant inbred lines. This you can say is the sequenced genome, and you can see that if it is red, it is the moneymaker, or which is compared to the sequence of Heinz, the sequenced variety. And red means that it's identical to the Heinz genotype, and green means that it's different from it. And then you can really clearly see that you have a nice representation of the different lines. In this way you can see the integration everywhere, and here you can see the picture we made by using let's say marker systems, AVLP or others, SNPs, and that looks very much the same. So at least that is quite comforting. So what you can do with this sequence information is you can very easily zoom in or home in on assembly artifacts of the Heinz genome sequence, and that is what we are currently doing. But at the same time you can also see what we were doing this for is to visualize the integrations in from pimpinellifolium in the Heinz genotype. Because you see here a part where it's always inverted, so you have the Heinz or the moneymaker sequence, and then you see this small part, and it's let's say all the time inverse with what you would expect. And this is due to the fact that Heinz already has some pimpinellifolium inversions. So the Heinz sequence is not a completely Solanum lycopersicum, but also contains some of the pimpinellifolium integrations. So in general, sequencing RILs and so on is a very good means of visualizing integrations. And because you can visualize integrations quite easily, you don't have to do cumbersome research like we did in the past for the TY1 resistance gene, where for a long time the breeders had difficulty in getting very good lines. They didn't know really where the TY1 gene was located. They had a lot of problems in integrating this trait, and this was due to the fact that it was more or less around the centromere oriented, and people didn't know whether it was the short or the long arm. Anyway, to make a long story short, if at that time they already had been able to sequence it, they would have known that they could select certain genotypes for using in the crossing experiments, and they would have given a better chance of integrating your trait in a much faster way. So the other part is that we sequence next to the recombinant inbred lines, we also sequence quite a number of wild species, and the idea was that we could identify then alleles which are, let's say, relevant to the phenotypic diversity across the entire genome and across the entire tomato clade. And this then is what breeders consider their treasure vault, and that's what we did. So we sequenced a number of different lines depicted here. So about 60 from the lycopersicum group, about 10 from the arcanum group, almost 10 from the this particular group, difficult name, and then from the last group also 9 or 10. So in total we had about 80, 90 different wild species and Solanum landraces. In this case there were about 50 landraces, and the average coverage was about 40x. So there were some initial analysis strategies in that without assumptions based, so on the K-mers there was a particular analysis, but also with some assumptions, and then we took the reference genome of Heinz, and then of course we would get information about SNPs, about evolution, etc., etc. And this looks a bit like shown here. So here you have the number of SNPs compared to Heinz. This is the lycopersicum group, and there you have the other three groups. And the more you get to this side, the wilder the material, and you can see that here you have a lot of SNPs, about 10 million, and here you have relatively few. But still quite a lot. And there is quite some variation in the lycopersicum group. So you can very easily see per chromosome the different integrations. So here you have chromosome 9, this is chromosome 11. You can see here, these are the landraces. So there are already some landraces which contain, for the tomato breeders, this is chromosome 9, so it contains TMV resistance. But you can also find that there is difference in the size of the recombination between the landrace and the integration from in this case pimpinellifolium. So you can select your line at best and look for the least linkage track. So that is quite useful. The other way of looking at the data, and this is a bit complex, is looking at haplotype diversity. The idea is always that tomato has no diversity or hardly any. It's very difficult to find markers, but it's the way how you look at the diversity. So what we did is we have here the 50-odd Lycopersicon genotypes, and here we have depicted 10,000 genes. So of the 30,000 genes, about 10,000 genes had one haplotype only, so we discarded those. Then we had another 10,000 more or less, which had more than 10 haplotypes. Those were also discarded because they might contain gene families and other things. And then you're left with this group, which contains between 1 to 9 haplotypes. And then you can see that there is indeed not so much variation shown here, but the further you go to this side, you still can find that maybe 4,000 or 5,000 genes, because these are genes, have a difference in SNP. So this can also explain why, although with normal systems you couldn't find so much variation, you still have a lot of phenotypic variation. So it makes clear that we can, let's say, zoom in much better on what is happening in the crop, and we can also use this kind of information. Because what you can do now is we have the information about the sequence from all these different lines, and of course we have the phenotypes. So you can look here, this is the Heinz genotype. We know that color is important in tomatoes, so the red ones, and we also know, luckily, that some of the genes of the pathway of the color. So in this particular case you have this gene on chromosome 3, it's a wild type. Then you can look at the yellow or orange ones shown here, and then you find that in these cases they're all a mutation which gives a yellow one. So this is the known mutation. But here you have a landrace which has an orange-yellow color, but the wild type gene is still present. So this might hint that another gene in the pathway responsible for color is mutated. So that might lead you to new genes which you could use, because maybe this gene is also better in combination with a good taste and flavor. Another example is the gene which is important for bricks in tomato. This was cloned already 14, 15 years ago from Solanum pennellii, and it was shown that 3 amino acid mutations or substitutions were responsible for the phenotype. So then you go to the sequence, and this is the sequence of the wild species, and this is only 275 base pairs. And lo and behold, you can see here the 3 amino acid substitutions, but you can also immediately see that the causal mutation must be this one, because here you see in quite a number of different genotypes that these mutations are also present, and these all are having a normal Brix. So this zooms in directly on the, let's say, the amino acid or the mutation which is present. If you then look, for instance, at Solanum calapacense, which is quite easily crossable with Solanum lycopersicon, then you can see that there are some more mutations present or in the exon, because this is the exon region, and here you have part of the intron region. So these might be then new interesting mutations to have a look into and see whether they have some effect on Brix. So in this way, the sequence can be used quite good and effectively in the breeding process. Well, what you would like to do as a breeder is you would like to go to a gene bank and give some information about genes. Then it should be possible, either you do it yourself, but preferably, let's say, a program will do it and gives you some information about which genotypes or which accessions, genotypes is better than accessions, you would have to order from the gene bank in order to find the diverse information about your different allelic diversity. And this is something we're setting up at the moment. This is an example in potato. We have been doing that now also for tomato and for mushrooms, and for some of the other crops we're busy. But what it actually does is it gives you information shown here. This is the gene bank information, passport data of accessions. We made some geographical information maps with it, and you get information about the different genotypes. We took from every accession 5 genotypes. Those were genotypes for a phylogeny map, and in this way you can, let's say, select from different clades of such a phylogeny the information. And then adding together the information on, let's say in this case, resistance against Phytophthora infestans, we made in some cases populations. The populations give a certain resistance segregation. You get information about the R genes which are present based upon their locus, but also in some cases the genes were cloned, and then you can go on and can just order this. And the way it looks like on the first page is that you have this kind of information where these are the wild species. You can click on these kind of things and get information about the place where they were collected. Here you have information about the resistance against 2 different isolates. You can see whether crosses were made, and you can also see whether genes have been isolated. And this is of course very important, because now you do not only get information about the potential R genes which are there, but you also get information to which clade they belong, and in some cases it is much easier and faster to have genes from specific clades or specific wild species which can be crossed much easier. If you look at this one, the RPI BLB2, one of my former bosses, he started in the end of the 50s to cross, and after 50 years there was a variety on the market which had this particular resistance there. If he had known at that time that a similar resistance would have been present in Stoloniferum, then he could have made directly the cross with the tuberosum, and it would have been possible to get this in let's say, a normal breeding cycle of 10-15 years for potato. So this kind of information is very important for breeders. So summarizing this part, I think it's important to have tools, pipelines, strategies for diploid and polyploid crops, and of course then you can think of what is important, especially in polyploids. Dosage calling of the different alleles is very important, but there are some other things which are also important. And in any case, you would like to have the lines of research which can aim towards an improved breeding process in the particular crop you are interested in. And that's why we set up the virtual lab of plant breeding. This is a public-private partnership. It's linked mainly to all kind of e-science domains, bioinformatics and ICT infrastructure. And the idea is that this is a dry lab infrastructure which must ensure that innovation in the plant breeding industry is being maintained. And the idea is that academic knowledge is then translated into let's say tools and methods which are relevant for both industrial and academic partners. And this is a bit how it looks like. So the idea is that together with the users, end users, we define problems. Then we look what is available in the world of tools, and these tools can be used directly or can be converted, or new tools have to be made. The idea is also that this is all with open software, and also using things like R for the statistic point of view. And then of course there are questions which can be asked for particular problems, and then answers will be given to that. And of course companies can also put their own tools or their own data, proprietary data, into this whole system. And the idea is that this will lead to, let's say, better visualization and analysis of all kind of sequence data. Because as a breeder it is a bit impossible, because you are not a bioinformatician, at least I'm not, to look at, let's say, 100 genomes which are sequenced, and then look at all the 100 genomes at the same time and have some idea about what kind of SNPs do I now have to focus on, which ones are important. So you need to have easy view systems for making that possible. You can also think about things like haplotype predictors. It would be very useful to incorporate identity by descent or identity by genotype information, structural variation, etc., etc. So we are at the same time developing specific for cross-pollinating crops, tools for cross-pollinating crops and self-pollinating crops, and for polyploids versus diploids, because the tools are not always the same. You have different questions in those different crops. Just take a So we started some years ago with what is called the VLPB1 program, and this was, let's say, a tool development at that time, and because there was no really large data file available, we decided to sequence 150 tomato genomes to have at least data with which we could start to develop those tools. At the same time, 100 brassica genomes were set up, and then this was continued in VLPB2 with 100 brassica genomes. That is still ongoing, but we also at the same time looked at more than 20 different cucumber diseases and did RNA-seq with resistant and susceptible genotypes in different time points, different interactions. So we got a huge amount of RNA-seq data, and that is all being analyzed and tried to be used in breeding as well. And now we are with VLPB3, where just recently a project on melon sequencing was awarded, and we are sequencing, just started with 50 tetraploid potato genomes. And one of the big issues in VLPB3 is also the data repository, because that is an issue. So we have a database, and of course there are a lot of databases, but this is one we use in Wageningen. And in this particular database you can navigate and can look at all kinds of different things. You can do data exploitation, as shown here. It will give you information about the different crosses which are made. It gives you information about the dots you see, what genotype it is, what the value is, and you can do many more analysis on the fly with this kind of information. And that is useful because it then gives you information about specific genotypes, what is happening with them. You can go back to the sequence and can look at the allelic composition and can try to relate it to each other. As I said, omics data is getting more and more important, and we're also looking into that, and this is an example of what we do with metabolomics data. And we try to, let's say, incorporate all that data, and this is an example of omics fusion program. And what this does is that it will use different statistical programs to predict what kind of metabolites are, let's say, highest in rank and most likely to be involved with a certain trait. And of course, if you find 5 or 6 times the same compound, then that might be a good indication that this is indeed a compound to be focusing on further. And that's something we do. You can of course do interval mapping, you can do genome annotation, you can go from the QTL region to your genome, you can go from the QTL region to the gene. There are some packages designed for that as well, marker to sequence, MQ2, you can find them. But all this makes that you are able to, let's say, assess the information which is available all around. A final one is shown here. This is Annotex, and what this does is that if you have, for instance, a region in the genome where you have a lot of genes available, then it might be able that you say, okay, I put in the genes and you get an answer about the proteins which might be important, but you also get information about different publications, not only in plants but everywhere, where it might give you a hint what kind of genes are most likely to be involved in the process you're looking at. So we have a flowchart for breeding, and that is shown here. So you do a lot of phenotyping and you do a lot of genotyping by whatever method. The dosage information is very important, especially in polyploids. You look at population structure, other things, and then in the end of the day you would like to end up with in silico prediction of your best cultivar. So that is what I call precision breeding. And the idea is that you can really design behind the computer the best genotype which will give you a good quality stable yields, etc. in different environments. And you can then, let's say, pick out your environment, the traits, and based on a cross between parents you know through and through, it will be able to predict what will be the outcome of that. And in this way, I think it will be much better to valorize all the investments which you are doing. Because it's nice to sequence a lot, but as a breeding company, yeah, of course you have to make sure that the costs are also recovered at some stage. So the idea is that you have very well characterized elite germplasm collections. You design your desired phenotype or genotype behind the computer, and you put your breeding goals as an input. Then you obtain the most effective breeding strategy starting with your elite material, and then cross, if necessary, genotype part or the entire offspring, doesn't matter so much. Yes. And then you select candidates which are similar to what you have designed, and then you can characterize them and maybe add them to the germplasm again, and then retrain your algorithms. So there are a number of different components which are important in this case. I point out ontology is very important because you need to really define very clearly what you are trying to do. But in this case, it is possible to, let's say, make the desired genotype with a much higher chance and in hopefully also less time. So this is where I would like to stop and acknowledge people who have been involved in this, people from plant breeding, but also from other departments, bioscience and bioinformatics from the University of Amsterdam, who are instrumental in the visualization, and then with the Center for Genetic Resources in the Netherlands. And this is where I would like to stop. Thank you.
We don't have much time, but probably we can take 2 questions. Yeah.
Good morning. I think this is the first presentation where I could hear polyploids. And this is an issue with polyploidy crop. And there are something that you mentioned is ontology. I think in many of the crops, the ontology has not been defined, although the CGIAR has now taken up that initiative to define the ontologies. I think that's where the bottleneck is. Because most of us are doing breeding, but even across the breeding, in different countries or different national programs or any institute, the ontology has not been defined. And I'm working in a crop called Dioscorea species, which is highly polyploid. And we are facing a lot of problem. Thank you for mentioning all those things. Thank you.
I had a question about some of the informatics management, the breed DB and the virtual lab. It all sounds very exciting. Could you just clarify a bit more about, are these meant to be web-based services, or do you download something to your computer and run it locally? How does it work?
So it's a combination. Most are web-based. Some can be downloaded. The BreedDB database has different areas. So there are some public areas, but there are also private areas. So it's a combination of all. But with most programs, we try to give the source code and information so that it can also be improved.
You can have more